All right, time to go teach. Hello and welcome to this next episode of Tutor Tutors, where we are continuing looking at the cell, and we are going to just go into part 2 of osmosis. In the last episode, we talked about how water moves based upon the tonicities that are present. In this one, we are going to be looking at how it impacts a cell. So first and only learning target for the day is we are going to explain the different cell types and how they are affected by these different tonicities. So remember, the 3 different tonicities are either hypotonic, hypertonic, or isotonic, and they are comparison terms based upon the solute concentration that is present. And how is that going to impact our cell? So let's look at it. First off, remember we have our cell, and it has this plasma membrane, and well, that plasma membrane is actually difficult for water to pass through, because water, despite the fact that it's really small, it is polar. And that polarity means it's hard for that water molecule to pass through that phospholipid bilayer. What that results is that our cells are actually going to use facilitated diffusion to allow for this water to pass through. So they are going to have protein channels. Those protein channels are called aquaporins. In other words, they're little pores for water, and they allow for water to move in and out of the cell. They allow for that water movement. So these are our pores that allow for the facilitated diffusion of water, which happens to be osmosis. So now our water can move in and out of the cell. Now, in an isotonic environment for our cell, we have an equal concentration of solute inside and outside the cell, which results in an equal concentration of solvent inside and outside the cell. And since the concentration of solvent is equal, our water is going to end up moving in and out of the cell. And it's going to do that in equal amounts. This is going to be at dynamic equilibrium. And the equal movement of water results in no change to the cell at all. It is exactly just stable. On the other hand, if we were to change this, and we're looking at a hypertonic environment, if we were to take an animal cell and place it in a hypertonic environment, the result would be that there would be a higher concentration of solute outside the cell, but it also would mean that there is a lower concentration of water outside of the cell. And so water would actually go from the cell out. The water would move out of the cell because there is a higher concentration of water inside the cell than there is outside of the cell. The inside of the cell was hypotonic compared to the outside, which was hypertonic. This would cause the cell to shrivel. The cell would shrivel and shrink, and we call this crenation. So as our animal cell shrivels because of the water that is leaving it, that is going to be called crenation. On the other hand, if we put an animal cell in the exact opposite type of solution, a hypotonic environment, well, that means that the water that is now at a higher concentration outside of the cell than it is inside the cell. And since the water is at a higher concentration outside the cell, it will then rush inwards. As it rushes inwards, it's going to cause the cell to swell, and then it will actually possibly overcome the plasma membrane and cause it to burst, and the whole cell is no more. It has burst open. It has released its contents out into the environment. It is no longer a cell, just like a broken balloon is no longer really a balloon anymore. It doesn't serve that purpose. This cell has ceased to exist. That process is called lysis. When an animal cell is put in a hypotonic environment, because the water outside the cell is at a higher concentration and rushes into the cell, it will cause the cell to lyse, which just means it splits open and it is no more. Now, some animal cells do exist in hypotonic environments, but what they have to do is they have to maintain homeostasis, they have to constantly pump water out. And the way to do that would be to use active transport. So cells, to maintain homeostasis, are going to use active transport to be able to exist in an environment that might not be the best for their survival. We can have animal cells that can live in a hypotonic environment, but to make sure that they don't lyse, they actually have to pump water out. As water passively just moves in and moves in and moves in, they are going to constantly be just pushing water out. Now, plant cells are a little bit different because plant cells, they have a cell wall. And that cell wall is going to change things a little bit. Now, first thing is, if it's in an isotonic environment, well, water is going to move equally inward and outward, same as what it was for the animal cell. It's a dynamic equilibrium. There's an equal amount of solvent concentration both outside and inside the cell, and so that means that we are going to have water move equally out and into the cell. On the other hand, if we took our cell and we put it in a hypertonic environment, in this case, again, that means that the water concentration is going to be lower outside of the cell, and so water will leave our cell, and as water leaves the cell, very similarly to the animal cell, our plant cell will shrivel and shrink, but it's actually pulling away from the cell wall. The cell wall doesn't shrivel and shrink along with it completely, and so we have a separate term for this. It's called plasmolysis. So the process of the plant cell shriveling is slightly different, and we call it by a different term. We call it plasmolysis. And this we can observe in a plant when the plant wilts. The reason that the plant wilted is because you don't have the cell pushing on the cell wall anymore. So because our cell has pulled away from that cell wall, our plant ends up wilting because of that process. On the other hand, if we had our plant cell in a hypotonic environment, higher concentration of water outside the cell than inside the cell, so the water is going to want to rush into the cell, that means that our plant will actually be really, really doing well. The large central vacuole will actually increase in size because of the amount of water that's rushing into it. We call this cell turgid. We refer to the pressure being pushed now on the cell wall as being turgor pressure. The cell doesn't burst because that cell wall is able to hold the cell together. It is able to maintain that structural integrity. Because that cell wall is there, the cell is going to be okay. It is not going to burst. And in fact, this is when a plant is the healthiest. When it is in a hypotonic environment, the cell is pushing very strongly, and this gives a plant its strong, rigid structure. So plants, they want to bring a lot of water in to maintain an environment that is hypotonic to their cells, so that way they have a lot of turgor pressure within their cells, making their cells turgid, so that way they are going to be nice, rigid, and strong. That is something that gives them some really nice structural support. If a plant is in a hypertonic environment, it is really bad as we have that plasmolysis taking place. And if it's in an isotonic environment, it will still be limp. It will still be not as rigid as it should be. It will be an unhealthy plant. So in summary of all of this, water, well, it uses that facilitated diffusion to enter and leave the cell because it uses those protein channels that are called aquaporins. And as it works out, the optimum tonicity for animal cells and plant cells, well, it's different. The animal cells, they work best in an isotonic environment. That's the easiest for an animal cell to maintain homeostasis in because it's not going to shrivel up and shrink, and it's not going to have the threat of actually bursting. So an animal cell, the easiest spot for it to maintain homeostasis is when it doesn't have to do any work at all. If an animal cell is in a hypotonic or a hypertonic environment, that means it's going to have to use active transport to bring water in or push water out. On the other hand, a plant cell would much prefer to be in a hypotonic environment. That's the optimum environment for it to function. And these cells, if they're not in the proper environment, they must use active transport. They must use energy so that way they can maintain the proper shape, the proper size, the proper amount of water either coming in or going out. Water will move passively. So if an animal cell or a plant cell requires to bring water in or to remove water, and water will not naturally do that, then the animal cell or plant cell must use active transport to maintain homeostasis. Otherwise, the plant cell or animal cell will cease to exist, and we don't have life right there. All right? So that is a little summary of how tonicities affect these two different types of cells. On that note, be awesome, stay awesome.